Okay, sorry. So what I'm going to be talking to you today about today is the principles of what we do, looking at identifying risk genes and how we do that. So why are we interested in genes? So for some diseases, termed heritable diseases or inherited diseases, they're caused by genes. So they're caused by a change or what we call a mutation in one of your genes. A prime example for this in dementia is Huntington's disease, so caused by a mutation in the Huntington gene. But most diseases, most complex diseases as we term them, so most dementias are partially heritable, which means that they are seen in an increased rate in families, or but that they're not determined by a genetic factor. So you can have an increased risk due to your genes, but you're not actually definitely going to get disease. So what we work at is to identify these risk genes and how they influence risk for disease, and longer term plan is to look at how the environmental cues as well work with these to increase risk. So how we look at it is that you have genetic and environmental risk for most dementias, and actually over time your genes and environmental factors increase to reach a threshold, and at that point you reach a threshold, then you go on to have disease. So we primarily work on late-onset Alzheimer's disease, which is classed as disease after the age of 65. So we know there's an increased risk in first-degree relatives, so we know that there's a genetic heritability. And looking at twin studies where they look at di or monozygotic twins, the heritability estimates for disease are up to 79%, which suggests that up to 79% of Alzheimer's disease is due to your genetics. Your risk for Alzheimer's disease is due to your genetics. So what we do with genetic studies, or the idea of them, is to help pinpoint disease mechanism, so to identify which genes are increasing your risk and which biology or biological processes these work in. We can then use this information. If we know which genes increase your risk, we can look at who is at an increased risk. So we potentially can predict who is going to go on to develop dementia or a specific form of dementia to allow earlier diagnosis. We can test the relationships between the genetic risk factors and the environmental risk factors to see that, for example, if you have a risk factor in one gene and you smoke, is that increasing your risk exponentially compared to just sort of adding together? And I also use the genes then to identify therapies and potential interventions. So looking at drug targets, or again, looking at those at a higher predisposition for certain environmental factors as well. So the basis of what we do, or the underlying principle, and this goes for all genetic studies, is genetic association. So essentially what this does is just look at the number of mutations within a gene in those people with disease and those people without disease. And this goes for all genetic diseases diseases that have a genetic risk component. And essentially all we do is look at whether the gene you have is, in this diagram, blue or yellowy, and how many of those are in cases and how many of those are in controls. So we term cases as people living with disease. So as you can see from this example, we would essentially say that the blue mutation is linked to disease because it is seen more in people with that disease than it is in a standard control population. So what we do is we do this en masse, we do it across the genome. So this is termed a Manhattan plot because it represents the skyline of Manhattan. And what we see is all these variants across the genome that we've tested for genetic association with disease, and then we get hits in certain genes. And this is statistically tested, essentially what I showed you on the last, cases versus controls, but doing it across the genome, and you get these nice Manhattan plots, which do look quite pretty. So this was the latest study that was undertaken 2 years ago now in Alzheimer's disease, and it shows how many genes we've got associated or as increasing your risk for disease. And what this allows us then to do is make these very pretty but complex pictures. And essentially what these are is looking at clumping these genes into different biology pathways. So we use something called the Gene Ontology Network, which is a database that basically puts all known information about genes and which biological processes they affect into a database. And we use this then to look at patterns in the data. So are the genes that we're identifying having a specific function? Do they clump together in a functional pathway more than we would expect by chance? And the same with the network on the right here. This is called a protein-protein interaction network. So it's known interactions between different proteins. Do we see the genes that we've got linking together and the proteins they encode, so that they go on to encode, linking together more than we would expect by chance? And this gives us a lot of information about what biology is going on in disease. So a lot of the work that we've done to date, identifying the risk genes, now links into the UK Dementia Research Institute. So there's an institute here in Cardiff. There's a number of others across the UK. The hub is in London, in University College London. And essentially the genes that we've found have gone into looking at pathways and different biological processes. So looking at different animal models and human cell models of disease and trying to understand what they're doing and how we can influence those with drugs and other things. So the only way this is possible is to have a research cohort. And we've been very lucky over the last 20 years that we've collected research participants from across the UK. So from Wales, England and Scotland. We had a small cohort in Northern Ireland as well, but the paperwork for that side of things was a little bit difficult. So we unfortunately didn't get too far. And CADR has helped us with that as well over the years. So part of our cohort was collected due to funding through the CADR project that we were involved with. And this is all the research centres that we had at one time across the UK. So we had a lot of sites that the girls over here and part of the previous team have helped coordinate and collect samples from. So it's been a huge effort. This has taken longer than I thought it would. So what we thought we'd do now is, what the girls are going to do, but obviously they do this rather than me, is talk you through what it's like to be one of those participants, one of those people that has come from one of those centres and how they the research actually works for them. But have we got any questions before we move on to that side of things? Because I know I've thrown quite a lot of information at you in quite a short space of time.
I just want to check that I understood. So there's 79% risk for genetics around Alzheimer's.
So 79% of the risk of up to, so it depends on the cohort and the study, but up to 79% of the risk for Alzheimer's disease is genetic. So some studies they're sort of high 50s, some studies go up to 79%, depending on, obviously there's different samples, different things. So it's come from twin studies, yeah. So, but there's obviously multiple twin studies. So some twin studies, I think the lowest was 56%, and then obviously different twin studies that come up with different percentages.
What are the assumptions then go to other non-twin siblings?
It essentially means that we know there's a large genetic component to disease. So obviously then that takes us, it basically gives us a good base to look at genes in relation to Alzheimer's disease. So other dementias have less heritability. So Parkinson's disease is 31%, I think it is. Dementia with Lewy bodies is sort of high 30s. Obviously different diseases have different heritability estimates. Andrew, did you have one? So there's a higher risk. Having a first degree relative gives you a higher risk of developing disease. The only way you would definitively get it is if the mum had a Mendelian cause for disease. So for less than 5% of Alzheimer's people with Alzheimer's disease, they have a causative gene. So there's 3 genes that have causative mutations in them. So if you have familial disease, if you have that mutation, you will go on to develop dementia, but that's less than 5% of cases. So for those that don't have the mutations in the sort of core disease causing genes, then you have an increased risk, but it's a risk, not a definitive diagnosis. You know, you're not going to necessarily go on to develop disease.
So then you just need to check what type the parent has.
So if the parent has familial disease,
yeah,
and the NHS do offer screens for familial, you ask for them, and they will offer screens for familial disease. Normally, people with familial disease have a very early onset, so normally in their 50s. So it's normally a very early onset for familial disease because it's a causative mutation. You do get some that are a bit later, but I'd say by mid-60s, if you develop it after mid-60s, you're very unlikely to have heritable, a Mendelian cause. So yeah, but at the moment, there's no NHS type testing for risk genetics, because to be honest, at the moment, it doesn't mean you're going to get disease. So, you know, if you live a very healthy lifestyle, you can have very high risk factors but not get disease. So, okay. Any others, or should we move on to and take Danny can step up and take over with this bit? Yeah.
All right, so my name's Danny. I'm one of the field team workers on a research study called Brains for Dementia Research. This is within the DRI at Cardiff University. Essentially what we do is we have a cohort of participants, some that have a diagnosis and most who do not. And we conduct annual assessments with them either in person or on the phone. And this varies from time to time depending on what their previous results were the year before. So this part of the session is hopefully an interactive one. So I'm hoping some of you will be able to help me out with this. But just to give you sort of an introduction into what we do, what those annual assessments look like, that's what the next few slides are. And I'm assuming I just pressed the arrow button. Okay. Lovely. So if you were one of our participants, this is what you could expect from us coming to you. We call all of our participants up to book them in for their annual visit. So we would call you the day before just to check that it's still okay for us to come and see you. We double check your address to make sure that we actually turn up to the right place, because that has happened before where we've gone to the wrong place. Often we arrive on time. Sometimes traffic can be a bit of a problem. Some of our participants live a little further afield, so there can be sort of a 2-hour travel time to get there. Some of the interviews that we do, so we collect quite a wide range of information, and that goes from medical information, sort of past operations, past illnesses you have, what medications you've been taking and what medications you're currently on. The memory assessments that we focus on are the mini mental state exam and the Montreal cognitive assessment. And I'll give you some examples of some questions we might be asking in a little bit. Other assessments we do as well as the National Adult Reading Test, and this is essentially where you have a large list of words that get increasingly more difficult as you go through them, and it's just to identify sort of, it's called an intelligence test, but we just sort of gauge to see sort of what people's level of understanding is and what people are able to read out loud, and we'll go through some of those in just a little bit as well. We also do a mental health assessment with the geriatric depression scale, just to assess how our participants are doing. And we do this test every single year as well, just to check in, make sure they're doing okay. And if they do score highly, we then have a small conversation with them afterwards to ask if they've spoken to someone about it, if friends and family are aware of how they're doing, and just give them a little bit more support and guidance if they feel that they need that at that moment in time. Alongside these sort of cognitive assessments, we also do some physical measurements, and this is just to gauge how they're doing physically. Are they walking? How fast do they walk? And I'll go into that as well in a little bit of detail shortly. So just to give you a little bit more information about some of our assessments, the mini mental measures thinking ability and cognitive impairment. So it focuses on 5 different domains of cognitive functioning. It's the most common test that's currently used when working with individuals who do struggle with their memory and in older populations as well. The main thing to know for the mini mental is if you do have a low score, it does not guarantee that you have mild cognitive impairment or are likely to have dementia at that stage in time. The Montreal cognitive assessment is, again, a rapid screening test for mild cognitive impairment. And this is a far more modern assessment. So it's got a higher sensitivity to detecting a mild cognitive impairment. And therefore, if you do score low on that, it can be indicative that you are struggling with memory and can help towards a diagnosis. Not from the research team because we're not clinicians, but something that we can also speak to our participants about if they are interested in finding out a little bit more about their memory and their health in regards to their brain. The National Reading Test is comprised of 50 words. As I said, it increases in difficulty. The words aren't phonetically as expected. So as they increase in difficulty, the way that you would read them are not necessarily how they're said. So we'll go over those in a little bit in a bit of time. And then the geriatric depression score, there are 15 questions. Some of these questions are a little sensitive, so we do sort of tread lightly with these questions just to make sure that we're not affecting someone's mood in that moment as well, because those who might not like to think about how they're doing and feeling, these questions can bring up a little bit of emotion in regard to that. So we are very careful in the way that we phrase these questions. And as it's a research study, our participants are not, they're not forced into answering every single thing. If they're not comfortable, we can skip those questions as well if they would prefer that. All right. So some example questions, and this is where you guys come in, okay? So I'm going to give you 3 words to try and remember, and at the end of this little talk, I'm going to see if any of you can remember the 3 words that I've given you. So the 3 words are apple, table, and penny. Some of you want to say it back to me just to make sure you've heard me all right. Perfect. Thank you so much. All right. So this is another example question. You guys can write this down or you can say it out loud. It depends what you're most comfortable with. This is one that a lot of our participants really hate answering just because it is a quick mental math. So yeah, if you could take 7 away from 100, which would be 93, and could you take 7 away again? Yeah, well done. And 7 again. Lovely. And again. Perfect. And once more, guys. Well, you guys got 4 marks there, so well done. All right. So this is one that we use on the marker test. Some of you might have paper in front of you. You can draw this if you'd like to. You also can just sit and listen. So what we ask is for them to draw a clock face, include all the numbers on the clock face, and set the time to 10 past 11. A lot of our participants struggle with this, and that's not necessarily because they're struggling with their memory or anything like that. Sometimes the numbers just get a bit funky. Sometimes the hands are put in the wrong place where it's sort of 10 to 11 instead of 10 past. So the points vary a little bit depending on what they've input for that. Lovely. All right. So ignore this because I've changed my mind. I'm going to ask you the 3 words in a little bit later. But in the meantime, I'm actually going to give you 10 new words to try and remember, just to confuse things a little. So I'm going to read out the 10 words. I'm not going to ask you guys to repeat it back to me because then it might get a little bit confusing in the room. So we're going to do 10 words. I'm going to do 1 short activity after that, and then I'm going to see how many words you guys can remember after that short activity. Okay, so the 10 words are cabin, pipe, elephant, silk, theatre, watch, whip, pillow, giant, and chest. Okay? Chest, like a treasure chest. All right. And could you guys tell me what the names of these 3 animals are? Lovely, thank you so much. The one thing we've noticed with our participants is they sometimes get confused between a rhino and a hippo. So those 2 get flipped quite often depending on how long they look at the picture and how quickly they want to get through the test. All right, so these are the, oh wait, go back. Can anybody remember the 10 words? Yeah. Well, yeah, nice. Brilliant. Brilliant. Well done, guys. That's very impressive. So often, just because there's so many words to remember, most people generally get around 6 or 7. That's if you're really good. Most people generally have 4 or 5 when they can remember it. So we ask them to repeat it twice throughout different assessments. I really struggled with it when I first did it. So when I was being trained, which was about 3 months ago, I'm still quite new to the study. I didn't do very well on that. So lovely. So these are the 50 words of the NART exam. So they go across, so called a deco aisle. And as you can see, as you go further down, they do get a little bit more difficult in pronunciation. I'm still learning some of them. I've got actually a little cheat sheet here in case any of you called me out on how some of the words was on some of the words there. But yeah, that's the NART. I could go through it if you want me to, but I'd rather not. All right, lovely. And then just before I go into the physical test, can you guys remember what the 3 words were that I said at the beginning?
Apple, table.
Lovely, well done. So the physical test, as I've mentioned already, we go quite into a bit of depth about what we collect with these. So with blood pressure readings, We make this optional just because a lot of people either don't like getting their blood pressure taken, or they're on medication that can make it a bit difficult to get a blood pressure reading, or it can be dangerous for them. But for those who are able to, we get 3 readings sitting down and 3 readings standing up. As you can imagine, this can be quite trying for some people. When I first received the blood pressure equipment, I forced myself to do it at home just so that I could have a bit of sympathy for our participants that do it. So I'm able to try and relate with them a little bit when they're having to go through that themselves. So we get 6, we hope to get 6 blood pressure readings in total. We then use a grip strength machine where they do 3 times on both hands, and I've actually brought it with me today. So if anybody wants to test their grip strength in this room, you're very welcome to. We can go around and see like who has the strongest grip or anything like that. We also do gait speed, so we set up sort of a distance on the floor and time to see how long it takes them to walk from one end of the tape to the other. And this is their normal walking speed. So sometimes our participants tries to test themselves to see how fast they can walk in that time. If they do that, we get them to do it a few more times, just so that it's a more accurate representation of how fast they can actually walk on a day-to-day basis. And then last but not least, we do a hip and a waist measurement. This is one of the newer things that we do in the study, and always brings out a range of responses from our participants, particularly the females, who are a little bit more self-conscious about where their waist is at that moment in time. So yeah, so would anybody like to test their grip strength in the room today? Examination. All right, so this is what it looks like. So you can see that there's a gauge, there's a face on it. When you grip it, the machine doesn't actually move at all. So when you're gripping as hard as you can, you don't feel any movement, but there's a change on the gauge. So I got 34 in that moment. Our average with our participants is anywhere between 10 and 25, I'd say approximately. So, and you can see how you're doing.
I'm not very strong.
So it's just to test how strong your hand is. Because then if you just hold there, for you one more time. Oh my gosh, okay. So Emily got 46 kilograms in grand final. With everything that can be 46 on the 15. Anybody want to say?
Lion. Lion.
Yeah. And I let go from the hard as you can, really. Okay. That is an average. You want to do that? Let go. Right. So you've got 30.
Can I ask, is it not an accident that the dial is set away from the person doing the squeezing? Because I kind of thought if you could see the gauge, that might kind of.
It might be
encourage you.
towards you, but it might be, but we should always flip this around so that you can see it. A lot of participants ask what their scores were, and often they want to do it against it. And we should let them do that because, you know, competition is a good thing. Would anybody else like to try?
To the right and left.
So we do both hands. Yeah, we do both hands. We do 3 on the right, 3 on the left, and then we know what the dominant hand is. And then let go, and then you can squeeze as hard as you can. Brilliant.
So what's
Let go.
24? That is a shoulders, so
That's it. And then squeeze as hard as you can. Brilliant. You want to let go. You've got 28 on that one. Well done. Sorry, there was a question. I didn't.
Yeah, it was actually asking what's the purpose, because some of the others, they make a lot of sense.
A lot of
but
sense.
What's the
Yeah.
purpose of that?
So purpose for the physical measurements, not quite connected to the research itself in regards to memory and cognitive impairment. The physical measurements is just to gauge how they're doing physically, how they're doing with their health. Some participants, sort of if they have Parkinson's, for example, are going to have far weaker strength in their hands than those who don't. So it's just to get a comparison from our participants to see how they're doing, what it can range from, and just to gauge physical strength, really.
And help with time. they've got quicker decrease on the gait, then it can suggest more of Parkinson's dementia than body type dementia. That's what they get. So this particular study is a longitudinal study where they donate their brain on death. So it's having the longitudinal follow-up as well for data avoids, which obviously looks at their well-being as well as their cognitive decline.
Okay. Could I ask about the reason? First of all, what's the purpose of using the depression scale?
Mhm.
And secondly, when it comes to the word-based test,
Mhm.
do you exclude the 20% of adults who have literacy problems, and and even within that, do you allow for educational levels?
We do, yes. So in regards to the depression assessment, that one, I'm not entirely sure the purpose of it being brought into the study. Unfortunately, I wasn't quite there when it was introduced initially as part of the assessment that we conduct. But I know that from that, the results from that assessment just gives us an insight into how they're doing. I know that loneliness is a really big thing for individuals in the older population. A lot of people struggle with isolation and with their mood, and because some of them might not necessarily have the social contacts to be able to speak to anyone and outreach for any help or support they might need in that moment, that assessment gives us the chance to check in, make sure they're doing okay, and if they're not, we can then be that person
So
for them,
that's like
provide them that support.
Secondary wellness, the central core.
That's my understanding of it. There could be more to it.
The other part of it is that in the early diagnosis stage, some people with dementia can look like they've got depression. So it's putting out. Some people are misdiagnosed with dementia when they've actually got significant depression and vice versa. And there's some evidence that depression in midlife has an increased risk of dementia in later life. So essentially, most of this is trying to get as many measures on a person longitudinally over time, so that we can investigate these types of. What's the age range of Parkinson's? So most of them are quite old now, so they've been enrolled. So the study can go back to 10 years.
Almost 10 years now, yeah.
So most of them now are in this. A lot of them are in their 80s actually, but most of them are 70s.
Got a few in the 90s as well.
Yeah, so they were specifically enlisted at the beginning to be sort of at the cusp of developing disease, so but not so young that it would take 40 or 50 years to be able to in the brain. So yeah, most of them were enrolled sort of late 50s, early 60s, but then a lot were sort of already diagnosed with disease to go forward. The study sort of was across the UK with one site out of six that studies every at the time. But it's getting the balance, it's getting the cohort and obviously potentially the brains donated to be used fairly rapidly while still maintaining a longitudinal cohort that we have longitudinal data on to be able to do various studies and look at various questions.
Sorry, to answer your second question about the reading test, we do take education into account. So one of the earlier sections, we ask how many years of full-time education they've completed, what the highest level of education is, if any like diploma, undergrad, A-levels, GCSEs, for example. I've yet to work with a participant who's not able to read, but in that instance, we wouldn't ask them to do that test because it would cause unnecessary distress in that moment, and we don't want that for our participants. They're volunteering for our study, they're doing us a favor in the long run. So we take no, not at all. So whatever they are able to give, that's what we'll accept. And if they're uncomfortable at any point in the assessment, we stop, move on, or we finish at that point. So we don't put them in any sort of distress. We don't like to make them upset in any way, shape, or form.
There is definitely a bias there with these types of studies that are higher educated and people that tend to have lower education don't tend to want to participate.
And the reason that we choose to start in the first place is something that stays quite consistent. So it's there as a measure that when people do one mark onto the pattern, even if they struggle with some of the other parts, such as the drawing, for example, from the memory test, their actual knowledge of how to pronounce words is really easy to do that at a young age. If you actually do that in the dementia, which is why we, even though it's really difficult, are very educated. Participants can do really well in it. They'll just continue to do really well in it, even if they don't.
Thanks so much, Tina. Really fascinating stuff, really. As a Welsh speaker, you know, 1 in 6 of people, or over 65 Welsh speakers, how does your study enable Welsh speakers to have the testing of those language they use? And how do you ensure that you sample contract sensitive of Welsh speakers or the population of Welsh speakers? Yeah. I don't think there's any. So the is run out of Newcastle. So obviously we are the only Welsh site. So there is definitely been a selection by a relation to people because we did have a Welsh speaker on the team, but she's better. So yeah, it's not funded well enough and funded to allow us to do it in those languages.
And the measures themselves.
The measures themselves can be into some maps. So they are translated. So they are usable in the translated form. We don't have it to deliver them. And to be honest, getting funding, even to translate after, you know, the sort of Welsh government legislative translation is still difficult.
We have had, my experience, I've got to the project for about 3 years. So we have a couple of families who look at all of their correspondence speak Welsh. So, you know, there's something now that is Welsh. Yeah, unfortunately, now that it's not Welsh, we're actually going to keep that like but it is unfortunate because, you know, that is especially if you have that all of Wales that we know we're going up ourselves for. So it's definitely a lot.
So when I do that, the word text going through. Thank you much. I booked the ticket on my mom.
Yeah.
But I assisted.
Yeah.
I mean, the only thing is it's really within a person, so it shouldn't, you know, so you know, hopefully it shouldn't change too much over time. But yeah, there's been the version more to the language or about information. So yeah, we looked at it well.
Got a question just here.
Yeah, a bit of both. So we're like comparing speak outputs and first yeah, that's what we're really interested in. The launching project is really interesting to see whether someone as they get older, as sportive as they always have, or what we're really supposed to started off with the forecast, but then they've gone on to that temperature, and we have a whole wealth of information of sort of every stage of that annually.
So how do you deal with someone's control person and then this looking like they how do you manage that?
So when we go on visits, we point out that something before we do anything to that. A lot of people, when we are worried about their memory, you know, the reason they take part in this project is often because with dementia, they're very interested in dementia. So when they start to be worried about their memory, you know, we're doing so we are what we want to say, but we have to be a bit relevant to that. If it's more sensitive, we can a bit more about it, and more if something we're concerned about, but we're not
So we've got a couple minutes left. I believe you've got a question for.
Would it be an longitudinal study and been around 10 years? How much more funding have you got, or how much longer are the study to keep going? At the moment, really well. So, the moment there is 3 years left on the current, and then the plan is obviously funded. So it's funded through ABB UK, which is a collaboration between Alzheimer's Society and Alzheimer's Research UK. So at the moment there is sort of verbal confirmation that it'll be another 5 years after that. It's funding landscape and things do change, but at the moment we're looking at another 8 years.
Just thinking in terms of, you know, the participants that they've obviously been involved in that some funding stocks, and this happening is going to first, and then like what did we see how we progress it?
Yeah, I mean, I suppose from a funder's perspective, the end goal has largely been the brain tissue reserve and although we're now recruiting cell lines from the participants as well from blood samples that we take. So obviously a lot of reserve has gone. The ideal would be we get to the point that everybody is donated, or at least attempted to donate. So that we did in the past, by the time the study finishes, I suspect that it will depend on numbers by the time we get to that point.
You don't actively,
We're not actively recruiting.
it's just that when it comes.
Yeah, so we're not actively recruiting for the study. Unless something changes in the next sort of year, I doubt we will. I think because what we've got now is the plan to see them through. But obviously it will depend on the type of kind of thing.
I'm afraid we've reached the end, but if you guys do have any more questions, just come chat to us afterwards. We'd be more than happy to have a conversation with you about any other questions you might have. Yeah, thank you so much, guys.
Yeah.